Yes, sir.
And hi, everybody. Welcome.
Hello, Derek.
I'm in Jersey. How you feeling today?
Better?
Good, good. Glad to hear it. Can you hear that?
Yeah.
I might have banging in the background.
Oh my God.
I'll put on the headphones. Hopefully it won't be too bad with those on.
Mm-hmm.
Oh.
The meeting, and I hope, yeah, the meeting is being recorded. So just letting you know that we're recording meeting, and I hope it's okay with everyone.
I take it that means yes.
Passive consent is always easy to obtain. So we just wait for 4 minutes. I think 5 p.m. is the starting time, and then we will start with the first talk of the department. I can see there are people who are not from JSPC. If you're from other school, whether faculty or student, please just drop in hi and school, the school you're from. It will be good to see who all is joining us. Just drop a small hi and the name of your school.
Hi, Sudiksha. Hi, Maria. Polkit. All right, we got both JSLH and JIBs here tonight. Welcome.
I am more, that makes me feel happy that all these email reaches to you and it doesn't go into other or a junk email directly. So great, everyone from JSLH, JIBS, and Global School, Law School for joining us.
Chinmay, welcome, Pearl.
Sharwangi, Sanskriti, are you from outside JGU? Oh, we have people also joining outside JGU also.
Yeah. Hi, Mahika.
Welcome, Pearl, Shreya, Sharwangi, who are not from JGU. Hi. So our social media channels are functional. Yeah. So again, a big thank you to all of you who are joining us from JSPC as well as other schools of JGU and outside the JGU. I know it's weekend. Many of you have a lot of things planned for the weekend. So thank you for taking time out and joining us at this first formal research event at JSPC. At JSPC, we are highly committed to advancing the knowledge for students and faculty and inspiring them to take psychology outside the classrooms and to real life events and real world. With this aim, we plan to have many more workshops, events, talks, conferences, dissemination workshop, which will help faculty and student alike to see how these concepts in classrooms can be demonstrated in real world and how they can be applied and understood. Faculty in Spotlights is one such initiative that we have started. It's an initiative where the selected faculty member will be coming and talking about a topic based on their area of interest, which can be from their clinical work or from their research work. This, we hope, will give students an opportunity to expand their knowledge, learn more about the research, but we also hope it gives faculty an opportunity to find common grounds for collaboration and keep on expanding their research work. So without further delay, it's 5 o'clock, so I will hand over the mic to Derick, Dean Derick. I think Derick will be in the best place to introduce himself, talk about his work, his professional experience, as well as the topic for today's talk. Over to you, Dean Derick. Thank you.
Thank you. All right, just to verify, you guys can see my screen, correct?
Yes, Derick.
Yes.
Perfect. All right, well, thank you very much for the introduction, Kanika, and I'm thrilled to be with you all tonight. So again, I am Dr. Derick Lindquist, and I'm the Dean of our new School of Psychology and Counseling. And so it gives me great pleasure to be the inaugural speaker of what will turn into a monthly seminar series where we have our various faculty come in and talk to students and anyone else that may be interested. So tonight, I've kind of taken a different tack with my talk. My plan is to really kind of give you an overview of my career. And to give you a little background on that, I attended graduate school at Yale University beginning in 1998, and I studied behavioral neuroscience. So I graduated in 2004. And if you would, please make sure you are muted, please. So I graduated in 2004. I did a couple of postdocs, and then I joined the Ohio State University. I worked in the psychology department there for about a decade. And in 2019, I moved to India and joined JGU. So I initially worked in the Jindal School of Liberal Arts and Humanities. And then earlier this year, I moved over to the Jindal School of Psychology and Counseling. So all told, I've done research for about 20 years. And so tonight is a bit of an overview to kind of give you at least a few examples of the type of work that I did as a behavioral neuroscientist. building towards a new theory or model of associative learning that I published last year. So we are going to cover a little bit of information, but I think everyone should be able to follow along. And with that said, let's get started. So, yes.
Yes, Derick. So just want to, clarify the audience. Will you be comfortable for them to stop you and ask question, or will you want, after finishing the sections or slide, you will stop and let them address their queries?
Yeah, I would prefer we hold off until the end, and then we should have about 20 minutes or so for questions.
Thank you, Derick. In the
Right.
meantime, you please feel free to drop your questions in the chat box.
Yeah, that's a great idea. And then Kanika, if you want to pick out a few exemplars, or we can have them ask in person. Yeah, why don't we hold off until the end, and I'm happy to discuss anything that anyone may have a question on. All right, so in psychology, specifically the side of psychology interested in the brain, one of the big revolutions of the last 60 years, so beginning in the 1950s and 60s, is a recognition that the brain is highly malleable, it's highly plastic or dynamic. Now, before this time, researchers knew that the brain could be modified, right? They were familiar with the idea of learning and memory, but on the whole, the brain was thought to be relatively static or unchanging. We've now realized, based on new technologies, that in fact, the brain is highly, again, plastic. It's literally being modified moment to moment, moment to moment. And I do apologize, just one second. All right. So while there are different types of plasticity or neuroplasticity, my goal tonight is to talk to you about one specific form of plasticity, and that relates to learning and memory. So the IT guys, is there a way we can avoid these popping up on my screen? So learning and memory. Learning is typically understood to be the result of some experience. It produces a relatively long-lasting change in an organism's behavior and or their thought. Memory is the outcome of the learning, right? So memory is what you store in your head. It's what you have access to at a later point in time. So when we take learning and memory together, which ultimately is, again, always based on your past experiences, anything before now is what your learning and memory is comprised of. So learning and memory enables organisms, including humans, to adapt to their environment. So some new situation comes up, you can apply and really sort of adapt and react to any different contingency. This is different than reflexes or instincts, which are both relatively fixed. Learning allows, again, a great deal of adaptability to respond to different contingencies, different situations, different circumstances. And moreover, as you learn about the environment, what you can do is you begin to make predictions. You say, okay, well, X, Y, Z, if X happens, I can make a prediction that Y or Z will also happen. So learning and memory evolved very early in our evolutionary past, literally hundreds of millions of years ago. So every organism on Earth today utilizes learning and memory, including single cell organisms to a more limited degree. Now, when we think about learning and memory, one of the great revolutions, again, beginning in the 50s and 60s, was a recognition that learning and memory, particularly more memory, it's not just this unitary phenomenon. In fact, there are different types of learning and memory systems, each of which depends on particular brain regions. So at the largest level, the way we can break down learning and memory is through what we call explicit versus implicit memory. So explicit is sometimes known as a declarative memory. And these are memories that you have conscious access to. So this can be your knowledge of the world. We call this semantic memory, right? So if you know that Bangkok is the capital of Thailand, that's a type of knowledge that you have in your head. It also includes episodic or autobiographical memories. And these are your personal memories, right? What you had for dinner last night, or the last time you took a trip with your family. Now, on the other side are these implicit or non-declarative learning and memory. For this, you don't need to be consciously aware that the learning is occurring. It's occurring in the background. You're going to retain that information regardless. So one of the simplest examples of this is what we call habituation. It means you respond less to a stimulus you encounter over and over. So imagine you move into a new apartment, right? You got all these wild dogs outside of there barking. So initially that keeps you up at night, that barking that you hear. But over time, you habituate, you respond less and less to the barking dogs until you get to the point where you hardly even notice them. Okay? Very simple. We also have what we call skills and habits. So this is sometimes referred to as procedural memory. This might be thought of as muscle memory, right? The ability to tie your shoelace, or if you're LeBron James, to shoot a free throw. So anything that you use your body for, we can see these procedural memories develop over time. Now, today I'm going to focus in on one specific type of learning and memory, and this refers to associative learning. So associative meaning you're associating together two or more stimuli. So there is two different types I'm going to talk about: emotional responses and skeletal muscle. Now, it's important to recognize that associative learning, right? Again, simply associating two or more stimuli or a stimulus and response. This is not a new idea. This idea was actually put forth by Aristotle, so about 2,300 years ago. And Aristotle, who we often consider to be kind of the first scientist, he was the first philosopher to really recognize the importance of empirical data, that is observation and measurement. So Aristotle proposed that if you have any two objects or any two stimuli that occur close together in time or space, they can become mentally associated in your brain. So as a simple example, imagine you see lightning in the sky. Thunder occurs, that really loud thunder causes you to flinch, right? You learn that lightning is followed by thunder. And so now in the future, when you see the lightning, you may wince or startle in anticipation of the thunder. So again, you've made a prediction. Thunder is always going to occur after I see lightning. And so your behavior has been modified as a result of that new associative learning. Now, there's two different types of associative learning. We have what's called instrumental conditioning and classical conditioning. So those in my class know the two. We just talked about this last week. So I'm only going to focus on one of these. This is really where the emphasis of my research is laying, which is in classical conditioning. So in classical conditioning, there is an association between two stimuli, a neutral stimulus and a biologically relevant stimulus. And that leads to the production of a new or learned response. So Pavlov, I'm sure almost everybody has heard of him and his dogs. So again, Pavlov didn't discover classical conditioning or associative learning. What he did is he designed a method, an empirical approach for studying classical conditioning. And this is really based on his ongoing studies into digestion. He actually won a Nobel Prize in 1904 for this work. But in his laboratory, what they noticed is, if we give food to a dog, this is our unconditioned stimulus, it leads to an unconditioned response. So by unconditioned, I mean reflexive. It just automatically happens. So if you present the food to the animal, the dog will salivate. Independently, we have a neutral stimulus. So this may be a bell that you ring. We call this a conditioned stimulus or a CS. Now, the bell is neutral, right? If you ring a bell in front of the dog, it doesn't care. It's not going to salivate. The bell doesn't mean anything. But here's the associative part, is if we pair the bell with the food, and we do this multiple times over and over again, the dog learns to associate the CS with the US, that is the bell with the food. And so now as a result of that association, I present the bell CS all by itself, and that leads to this brand new response. We call this a conditioned response. This is the learned behavior, the learned response. And so in my research, I've been really interested in trying to understand, how does that occur? How do the CS and US become associated? And how does that lead to this new response, this conditioned response when I present the CS all by itself? Now, when we think about learning and memory, there's actually multiple steps associated with it. So, of course, at the beginning, you have to bring in new information, right? So in the example with Pavlov, the dog has to hear, that is, it has to sense and perceive the bell, and then be able to eat and taste the food. So the incoming information, when it initially comes into your brain, we call this encoding, this laying down that new memory. But that new memory is often only temporary unless you store it or you make it permanent. This storage is responsible or due to a process called consolidation. Okay. Now, at a later point in time, your brain, once it's stored, it can retain that information and retrieve it and use that information for future behavior. So this process is called retrieval. So if we think of Pavlov, again, they encoded the CSU association, the tone with the food. They consolidated that over the time. And then at some later point, we present the Bell CS all by itself. The animal has learned that is predictive of food, and it therefore salivates. It shows this new conditioned response. So the question I want to start with is, how does the brain associate the CS in the US? Well, to understand this, we need to dig into the brain a little bit, okay? So I'm going to talk to you all a little bit about how neurons operate, and specifically the connections between neurons, which we call synapses. So this idea of trying to understand behavior and relate it back to changes in the brain, that is the nexus. That is the idea behind behavioral neuroscience, which is really a psychology-led discipline. So most behavioral neuroscientists are interested in research, trying to understand new information in order to build up our knowledge of the world. Most behavioral neuroscientists, not all, but most, including myself, work with animals. Simply because if we want to understand how the brain works, we need to manipulate the brain. So as behavioral neuroscientists, we may modulate the brain. So we may induce a lesion. We may inject a drug that damages the brain or inactivates it, makes it so it doesn't work as efficiently for some period of time. We can measure the electrical activity in the brain. We can look at different structures and assess the cells, the neurons that make it up, or the proteins or the genes. Okay? So my point is, there's lots of different techniques that are employed by behavioral neuroscientists. Now, for all of them, right, I did not work with rats for 20 years because I necessarily care a lot about a rat or how its brain operates. I did it because most every brain structure in your brain or my brain can also be found in the brain of a rat. And what we know is that the mechanisms, how, for instance, learning and memory is encoded in the brain, the mechanisms we uncover in the rat brain are very similar and in many cases identical to what we find in the human brain. So behavioral neuroscience as a field is really trying to understand that relationship. How does your brain control not just your behavior, but all these psychological phenomenon that as psychologists we are interested in? So if you will give me 3 or 4 minutes, I'm going to do a very brief little tutorial on how neurons operate. Okay. So first, we have to recognize the complexity of the human brain. On average, it has somewhere between 80 and 100 billion neurons. So neurons are brain cells. There's more than 100 different types of neurons that we have identified to date, but pretty much every neuron operates in a similar way. Okay. There's the top of it. The top is these little finger-like projections that we call dendrites. That's where the receipt of new information comes in. That information is filtered down to the cell body, and then ultimately the output of a neuron occurs through the segment we have here called the axon. Okay, the axon will form a connection or a synapse with a different neuron. And so the synapse, again, is how, or the location where one neuron communicates with another neuron. It's how they talk to each other. And again, to keep the complexity in mind, if you have 100 billion neurons, each of those neurons has between 10,000 and 200,000 synapses on average. It's just amazing in terms of the computational processing scale here. The brain, the human brain specifically, is by far the most complex that we know of in the entire universe. And so we understand a lot about how the human brain operates, but I often say we're still, it's like preschool or kindergarten. We understand a lot of the basic features, but there's still an awful lot that we need to learn until we truly understand how the brain can control us as people, and again, all those psychological phenomena. So how does one brain cell talk to another brain cell? Well, if we look at this axon segment, an axon or the neuron itself will generate something we call an action potential. Again, I suspect that most of you have probably heard of this. An action potential is nothing more than an electrical pulse. Okay? So if we look over here on the top, that little blip that you see traveling down the axon, that's an action potential. It's just a little electrical blip. Now, when it reaches the bottom of the axon, we have all these little connections. We call it an axon terminal. So that electrical action potential causes the terminal to release neurotransmitter. And again, most everyone's heard of neurotransmitter. Serotonin, dopamine, right? These are all different types of neurotransmitter. So that neurotransmitter is released into the synapse. Okay, the synapse is the gap here. It's what separates one neuron from another. So on the opposite side, and we call this the postsynaptic neuron, it's just the opposite side from where the transmitter is being released, we have receptors embedded into the terminal. The neurotransmitter binds to and activates a receptor. And what does that do? Well, it causes an electrical response on that second neuron, the postsynaptic neuron. So here's the action potential coming down this yellow axon. It causes neurotransmitter to be released. And that in turn results in the increase of electrical charge on the second neuron. And that's what this little signal here is meant to emphasize, okay? Little electrical charge. Now here is an actual image of a synapse. Okay, here's the axon on one side, here's the dendrite on the other. This little thing sticking up is called a spine. But that gap, that meeting place between the two is the synapse, all right? And that synapse is ultimately the place where learning and memory is embedded and encoded. So if you've understood me to that point, I now want to move on and talk about some of my own past research and how we've built on this existing knowledge. So one of the really important ideas, again, this really came about in the 50s and the 60s, is a recognition that different parts of the brain are responsible for different functions. Now, again, for most of us today, even people that don't know a lot about the brain, that seems pretty intuitive, pretty simple. But before the 50s and 60s, it was thought that the brain was essentially unitary. Any part could do anything. All right, we began to recognize throughout the 1900s, and particularly the second half of the 1900s, that no. In fact, we have all these different brain regions, each of which is responsible for something different, a different function. So I'm going to focus today on three particular brain regions. One is called the amygdala. It sits down here at the bottom part of your brain. The hippocampus, which sits right next to it. And then the structure at the very back of your brain. This is called the cerebellum. Now, the reason I'm focusing on these three brain regions is because each of them is responsible or controls a particular type of classical conditioning. So in other words, classical conditioning can occur in multiple areas of the brain. It depends on the type, the procedure of the classical conditioning that you do that determines which brain region is engaged. So the amygdala is involved when emotion is included. The hippocampus is required when there's some sort of contextual or place information that is encoded. And the cerebellum is needed when you're associating a sensory input with a specific motor response. So to begin, let me introduce the amygdala. It's this green structure that you see here at the rotating brain. So the amygdala, again, is responsible for emotional classical conditioning. We call this fear conditioning. And in fact, the amygdala plays a big role in any emotional responding that a person is engaged in. So if you are happy or sad or angry or afraid, your amygdala is involved. But a lot of the research in this field is focused on fear. And the reason is fear is easy to assess in humans and animals. Okay? So here's the paradigm. Again, simple classical conditioning. We pair a tone, which is our CS or conditioned stimulus. We present that for some period of time, let's say 10 seconds. At the end of the 10 seconds, they now get a shock. Okay? And you can see here I'm talking about rodents, or a mouse or a rat. This shock we apply to their feet. This is our unconditioned stimulus, or the US. Now, I want to make clear this doesn't hurt the animal. It's a little annoying, a little mildly painful, but there's no permanent damage. They just shock their feet. So they sort of jump around and dance, and it's not pleasant, but there's no long-term harm to the animal. Okay? So let's imagine I present the tone and then the shock, and then the tone and the shock, and I do this 10 times. Okay? So the question is, how and where do the CS and US become associated? Well, first, it's important to recognize that the amygdala is involved in this learning because, again, the amygdala plays an important role in emotions, particularly negative emotions like fear or anger, right? So the body can be activated. This is known as the fight or flight response, okay? And again, I bet most of you have heard of this. So the fight or flight response, if you see a snake, right? You're outside taking a walk in the evening and you see a snake, which has happened to me multiple times here where I live, right? What happens? That visual information is processed and is relayed to your amygdala. Your amygdala goes, oh, wow, look, potential danger, potential threat. So the amygdala turns on or activates that fight or flight response. Okay? Your heart rate suddenly starts to beat really fast. You start breathing fast. You start sweating. Your body prepares itself to either fight or flight, to run away. Okay? So if you ever hear a loud noise and you're startled and suddenly your heart rate is beating really fast, again, that's due to the fight or flight response. And the amygdala is the brain structure responsible for turning that response on. So when we think about the association of the CS and the US, it occurs in the amygdala. Now, the first thing we need for this to occur is the sensory information has to be relayed to the amygdala. Okay? So again, you hear a tone, that information is initially relayed to your ear. In your ear, in a structure called the cochlea, that auditory sound is converted into a neural signal, which is nothing more than action potentials. So you have a neuron that starts firing action potentials or spiking. That activates another neuron, which relays it up to the brain. And then in the brain, that information is ultimately going to be relayed to the amygdala. Okay? Same is true for that foot shock, right? The sensory information is relayed from the foot up into the brain and then ultimately to the amygdala. So in the amygdala, we have neurons, individual neurons that are receiving sensory information related to the auditory CS, the tone, as well as the aversive US, the foot shock. So here's my little cartoon for you. So let's imagine here's that CS, that tone information coming into the amygdala. Okay, here's the amygdala itself. And we're going to record the action potentials from one of those neurons. And here's the output from the amygdala. So these little dashes that you see, those are just action potentials. Now, I want to note here that initially the CS, when we first present it, it's just a random tone. It's completely neutral. It doesn't mean anything. So it has a very low level response in the amygdala. The amygdala doesn't respond. It doesn't care. There's very little activity. Okay? But now we're going to pair the CS with the US, the tone with the foot shock. Now, that foot shock is emotionally arousing. Right? This animal just got shocked. It's jumping around and going, what the hell, right? Somebody just shocked my feet. It induces a fear response in the animal. So that's information related to the foot shock US is also relayed to the amygdala. And the really thick lines here indicate that this is a really strong signal. And when we look in the amygdala, you see all these little dashes. That means there's a lot of action potentials generated in the amygdala because of that US. Okay, it's a very strong stimulus. Now we're pairing that strong US with our weak CS. And ultimately what's going to happen now is that we're going to transform that CS signal. It becomes stronger. And ultimately that strong CS signal, which looks like this, right? So initially the CS evoked this much activity in the amygdala. Now, after the pairing with the US, the CS all by itself elicits a lot of activity in the amygdala. So much so that the amygdala now begins to fire its own action potentials, and it activates other areas of the brain, areas that are responsible for the conditioned response. Okay. So again, if we go back to Pavlov, that conditioned response would be every time the tone CS is presented or the bell CS is presented, salivate, right? The animal causes the animal to salivate. That is the conditioned response. Now, what's actually happening here is a process called synaptic plasticity. Okay? And this is the plasticity that I really want to focus in on today. It's a change in the strength of the synapse. In this case, specifically those synapses in the amygdala that are activated each time that tone CS is turned on, right? The tone is flipped on, the animal perceives it, and that CS activates the amygdala and neurons within it. So initially, the CS and its strength at the synapse was relatively weak. But by pairing the CS with the US, we increase the strength of the synapse. That is, the synapse undergoes something called long-term potentiation. Okay? Now, if we wanted to weaken it, we would undergo long-term depression. We can just ignore that for the moment. Let's just focus on strengthening the synapse. So potentiation means to enhance, to strength. So what we've done is that we've strengthened the synapse. All right. Now, again, folks, I want you to understand, if you don't understand every detail, that's okay. This really is a lot of information that I understand. I want you to kind of get the gist. I want you to kind of flow with it, and you kind of have at least a basic understanding of what's happening in your brain when this associative learning occurs. Okay. So let's go down one step. And now we're actually at the level of individual synapses. Okay. So this neuron here on the right is the amygdala neuron. This on the left is the CS signal coming into the amygdala. Okay. Now, initially, again, the CS is very weak. So it has a very weak response in the amygdala. That is, it causes very little electrical charge buildup. And that's what these three little plus signs mean. Okay, very little change in the amygdala when that tone CS is turned on because it's neutral. It doesn't mean anything. Okay, so the amygdala, there's no output, there's no activation. Excuse me. But now we pair the tone CS with the shock US. Okay, so they're actually overlapping in time. At the very end of the tone, we present the shock. And so the CS and the US are overlapping in time. We have a receptor in the brain called the Nmda receptor that recognizes that coincidence or that association. It recognizes that this weak input is occurring together at the same time as the strong input. Okay, that is, the tone is being activated in your brain at the same time the shock is being activated. So that leads to a change. This Nmda receptor sends out instructions, and it says, okay, here's what we need to do. We need to add more Ampa receptors. Okay? And again, the details here don't matter. But the idea is if you look over here at these synapses, and then we compare it to the ones over here, you see how there's more of these brown receptors in there? These brown receptors are these Ampa receptors. And what do? Well, every time an action potential occurs when the tone CS is turned on, it causes a buildup of electrical charge in the amygdala neuron. So after the CS and the US have been associated, more Ampa receptors means a bigger response. More electrical buildup means more excitation. So that amygdala neuron becomes so excited that it begins to fire its own action potentials. That is, that amygdala neuron that initially did not respond to the CS, now it does. It begins to fire. And that firing activity is our conditioned response. That's how the amygdala learns to associate the CS and the US, and it begins to emit or generate this newly learned or newly acquired conditioned response on the part of the animal. Okay. Now, again, if that's a little confusing, that's okay. No worries at all. Here's the idea, though. When you have these two stimuli associated, the CS and the US, all that's happening in those synapses is that we're turning up the volume, okay? It's like turning the volume switch. We're making those synapses stronger. So initially, the weak CS is now a strong CS. It elicits a bigger response. We've turned up the volume. And now the amygdala, which initially did not generate its own output or its own response, now it does. And what's the response in fear conditioning? Well, there's actually multiple responses that we could measure. Because again, this is part of the fight or flight response. This is that emotional arousal associated with the fight or flight. So we have changes in heart rate and blood pressure. If you're a rodent, you emit these vocalizations that are above the threshold for humans. So we can't hear it, but we can use equipment to hear it. So these are called 22 kilohertz vocalizations. But the most important part, a rat or a mouse that is afraid, it freezes. Okay? It just becomes immobile. We can measure the amount of freezing in an animal, in a rat, very, very easy, okay? So here's the idea again. I pair the tone CS with the shock US, and I do that 10 times. Okay? I wait a day, and then the next day I come back and I present the tone CS all by itself. And the question is, did the animal learn to associate the CS with the US? If it did, when I turn on the tone, the animal becomes afraid and it begins to freeze. So I simply measure the freezing behavior, and that is my dependent variable. That is my measure of learning. That's how I know if the animal has successfully associated with the CS and US or not. Now, how do we know that the amygdala is involved? Well, this is actually an example of my own work. So this was done back in the early days of graduate school. We damaged or lesioned the amygdala, or we put in a drug that interfered with Nmda receptors, and sure enough, the animals were no longer able to learn. Okay? So the next paradigm I'd like to introduce is contextual conditioning. Contextual conditioning just refers to a time, a place, or a location. In humans, the hippocampus is really critical for that declarative memory I mentioned earlier. So your knowledge of the world or your own personal autobiographical memories. It's very difficult to assess that in a rodent, but we can assess what we call context memories very easily. So if you want to think for a moment, just close your eyes and think about your living room, right? So what we know is that people are able to build up a memory of their living room. So you take all this spatial and sensory information and you kind of pile it together and mold it, and you make this unitary context memory. So if you think about your living room, you don't think about just the lamp or the couch, but rather you can see the size of the living room, you know all the objects in it, and their spatial representation to one another. So this is analogous to an autobiographical or a semantic memory, where with a context memory, the rat in this case gets put into a chamber, has a particular size, it has grid bars on the bottom, it may have a light, it may have a smell and odor associated with it, it may have a sound. So again, the animal can recognize a particular location or a particular context. Now, when we build up these context memories, it actually occurs through a process called a memory engram. So I talked about one neuron being modified, the synapses being modified as a result of pairing the CS and the US. Well, a declarative memory or a memory engram is nothing more than a bunch of neurons that are all being activated simultaneously. They're all being strengthened together. So these 6 or 7 neurons in this little cartoon, they form a memory engram. So what is an engram? Well, if we look at all the neurons in your brain, if you remember your walk to class this morning, right? That's your first engram. That's going to activate a certain pattern of cells. The red ones here are the pink ones. If we asked you to think of another memory, maybe eating lunch, that activates a different pattern of cells here in gray. Okay? So a memory engram is nothing more than a collection of neurons that are being activated, and together their activity forms that new memory, be it a context or a declarative autobiographical memory. So when I was at Ohio State, part of my work involved looking at the deleterious effects of alcohol early in development. Okay? This is a disease known as fetal alcohol spectrum disorder. And I suspect everyone out there knows that pregnant women should not drink alcohol, right? It can lead to these impairments in their children. Physical impairments in terms of their brain and how it develops, as well as physical impairments in their body. But it also has functional impairments. Their attention is altered. Their responding to sensory stimuli is altered. And their cognition is altered, particularly learning and memory. So I wanted to investigate this at Ohio State. And one of the ways I realized I could do this is by developing an animal model of fetal alcohol syndrome. Okay? Now again, just real quick, the idea is that I administer alcohol to a newborn rat. Okay? Their brain at this point in life is comparable to the 3rd trimester in humans. So I can give them a milk solution that contains ethanol or alcohol, a low dose, a middle dose, or a high dose. Okay? That's my experimental group. And then I have control groups that don't get any alcohol, and I can look at the relationship between them. So we did a number of experiments at Ohio State looking at these memory impairments in animals exposed to early life ethanol. The first here is called the immediate shock deficit. And the idea is simple. If I put an animal into one of those conditioning chambers that I showed you earlier, but very, very quickly, within just 5 to 10 seconds, I shock it and then I pull it out again. Okay, that's the immediate shock. The next day I put it back into that same chamber and I simply ask, does the animal freeze or not? If it freezes, I know it successfully associated the context or the memory of the context with that foot shock. If it doesn't freeze, no association occurred. So initially with this immediate shock, the animals don't learn to associate the shock with the context, right? It's the same as if I walked you into a brand new room you've never been before, I shock you and then I throw a hood over your head. You're not going to remember it. It takes time to kind of look around, to explore this new environment in order to form a memory that you can associate with the shock. So all of our groups show the same low level of freezing. But now we change it. Before that immediate shock, I now introduce the same context. I don't give the shock, I just put the animal in the context for 5 minutes. The animal wanders around and it forms a context memory, a memory of that specific context. Okay. So the next day I come back, I put him back to the same conditioning chamber, and then 10 seconds later, I give the animal a shock. But because it already has a memory of that context, it looks around and it goes, oops, the same smell, or oops, I remember this grid floor. Even sampling a tiny bit of that context is enough to reactivate the whole memory. So the context memory can be associated with the shock. And when I come back the next day, I find that the animals freeze a lot. At least my control animals that did not receive any ethanol did. Here are my animals that received ethanol: low dose, medium group, and high dose of alcohol. The lowest level of freezing is in my high dose of alcohol group, meaning they had difficulty. They could not either encode the context memory or they were not able to store it so it was available 24 hours later when I give them the immediate shock. So how do we test that? Which stage is impaired? Is this an encoding deficit? They can't encode that new context memory, or they can encode it, but they can't store it or consolidate it. Okay. So here's the design that we came up to. We still do the context pre-exposure. We put the animal in this chamber for 5 minutes, but now the duration between the pre-exposure and the immediate shock is going to be altered. One group of animals, we present the immediate shock 2 hours later. The other group of animals, we wait our standard 24 hours. Okay. Now, we knew already, based on our prior work, that at 2 hours, the animal has a short-term memory available to it. That is, no consolidation is required. At 2 hours, that memory is still present. You only need the memory consolidation after 24 hours. So what we found is, with a 2-hour duration, our animals that were exposed to ethanol, they performed as well as our control animals. There was no deficit. Okay? So that's this group up here at the top. All 3 of these little figures are the same. So what that tells us is our ethanol animals, they can encode a new context memory. It's available 2 hours later. But by 24 hours later, that memory is gone. It's absent. And we see that here at the bottom. So these are my ethanol exposed animals and my control group at the top. So what it suggests is that our ethanol animals, again, the ones who get ethanol early in life, they can encode a new context memory, but they can't store it into their long-term memory. They cannot consolidate that new memory. So based on this, we had a pretty good understanding of what the problem was. When we administer alcohol to these animals early in life, it leads to a memory consolidation deficit. And over subsequent years, we did a lot of research trying to understand exactly what happened. But ultimately, we figured that the ethanol or the alcohol is leading to an inflammatory response in the brain. Okay? Just like if you cut yourself and your cut becomes inflamed due to your immune system, well, we have the immune system and there can be inflammatory responses in the brain as well. What we determined is that neuroinflammation as a result of the ethanol, it disrupts the development of the animal's brain. So we came up with a very simple experiment. What if we simply give the animals an anti-inflammatory, such as ibuprofen, every time we administer the ethanol? And that's exactly what we did. We would give the ethanol, and then 2 hours later, we would give them some ibuprofen. We'd give them the ethanol, 2 hours later, they got ibuprofen. And we did this for 6 days. The animals grow up, and when they were adolescents, we did the similar task to what I just showed you, but there's no impairment. These animals that were exposed to ethanol, as long as they got the anti-inflammatory drug, that seemed to rectify or diminish the severity of those effects of alcohol. So the animals essentially showed a recovery. So the reason I'm talking about it shows that a lot of my work is interested in knowledge for knowledge's sake. But as I went forward, I became more interested in trying to apply the knowledge that I know, right? I understood how a rodent encodes and stores or consolidates memories. So by using that information, I can then begin to understand a particular disorder. In this case, fetal alcohol spectrum disorders, right? And so by using that knowledge or applying that knowledge, we were able to better understand the etiology of fetal alcohol spectrum disorders. Why does early life exposure to alcohol or ethanol, why does that lead to these disruptive effects in both physical development and cognitive development? All right, so the last paradigm I'd like to talk to you about is something called sensory motor conditioning. It's still classical conditioning, but now we're talking about a different area of the brain. This is called the cerebellum. It sits at the very back of your brain. And this area of your brain is involved in movement or motor control. So this type of learning means you have a sensory input which instigates or elicits a very specific motor output, a very specific motor response. Now, the type of conditioning I would like to talk to you about is something called eye-blink classical conditioning. I did a lot of this work as a postdoc. Okay, so in this, it's again classical conditioning. It was first developed in the early 1900s in Germany and then exported around the world. So this photograph on top is from the early 1900s. This is a modern example right below it, the picture of the girl here with this rig. But most of this, the behavioral neuroscience research was done in rabbits. Okay? So here's the idea. I take a tone CS, and again, I simply present it to the animal. The tone CS is usually fairly short, so maybe a half a second. But at the end of that, I have a little puff of air that I direct at the eye of the rabbit. Okay? So in the girl here, she hears the tone, and then there's a hollow tube here that will direct a little air puff towards her cornea, towards her eye. Now you may wonder, does that make you blink? It does. If you have a sibling or a spouse sitting next to you, have them blow in your eye, you'll see it's not very pleasant. Okay. So again, another type of aversive classical conditioning where the animal learns to associate the tone CS with the air puff US. So now after they've learned the association, I present the tone CS all by itself. And what does the animal do? Well, it's learned to blink. So it's learned to produce an eye blink conditioned response. Now, why is this advantageous? Well, let's look at the animal up here, right? The tone comes on, and now the animal learns to blink just before the air puff. So if the animal is able to blink, it's adaptive. It blocks the air puff because the eyelid is now closed. So here's what happens. You learn to blink, right? Everybody knows a blink. So here's my conditioning paradigm. I present the tone CS, and that's this little orange line here. So this is when the tone is actually turned on. Now, at the very end of that, I present that air puff US, okay? A little puff of air directed at the eye, and you can see they overlap together. So when the tone turns off, the air puff turns off. So this is the dash line for the CS, and when it comes on. This second dash line is when the air puff US comes on. This green line here is a measure of the eyelid activity, okay? In a rabbit or a person, I can measure the muscle that's responsible for your eyelid closing. It's a muscle called the Orbicularis oculi muscle, okay? Very easy to do. So this big increase that you see here, as the green line goes up, that means the eyelid is coming down, okay? This is the blink to that air puff US. Now, initially, that tone CS is neutral. It causes no CR. But as we go across training, day 3, day 5, by the time we get out to day 5, this increase in the green, this is, again, that just signifies that the eyelid is closing. This is the eye blink conditioned response. So now in an animal that has learned this well, they have learned not just to blink, but when to blink. That is, the animal has learned to blink just before that air puff US. So that blink CR is now adaptive, and I block my eye. If my eyelid is closed, I'm no longer bothered by that air puff US. Okay, so again, very predictive. The CS is very predictive, but it leads to this adaptive response on the part of the organism. So again, I have done this work for knowledge's sake, simple research, but I've also done it to apply this research to understand the cerebellum of the animals that were exposed to alcohol. So again, coming back to this FASD rodent model. And just like those animals exposed to ethanol were impaired in context conditioning, they're also impaired in this sensory motor or this eye blink conditioning. So over here on the right, if I just measure over training, how many of these conditioned responses does the animal emit? The top line here are my control animals that get no ethanol. The bottom line here is my high dose ethanol. So this just shows that they're impaired. They don't produce as many eye blink conditioned responses. But we also know the response itself is different. So again, this is an EMG response. So up at the top, this is where the eye begins to close. This is maximal closure, and then the eyelid begins to open again as you move down the right side here. So in my control animal, there's a really big trace here. And the maximal closure occurs almost exactly when the US occurs. That's the dashed line, okay? So what this means is this control animal that did not get ethanol, it has a really strong, robust blink, right? That lid is really closing hard. And it's almost perfectly timed. So the eyelid is closed at the point the air puff US would normally occur. This is the blink on the part of my ethanol exposed animal. So we notice that it's smaller, right? The amplitude is smaller. So this is not a strong blink. This is like your eyelid kind of fluttering closed. It's not a very strong blink at all. And we also know that it's not very well timed relative to the air puff. So the maximal eyelid closure actually occurs after the air puff has already occurred. So my take home here is that the alcohol in early development, it leads to impaired development of the brain, and it alters the learning and memory capabilities of these various structures, the hippocampus as well as the cerebellum. Now here's where I've been working to, and I hope everybody's been able to follow me so far. So as I got towards the point in my career where I began seriously thinking about moving overseas, okay? Back in 2018, 2019, I began to reflect over my career. And as I did, I kind of recognized some patterns begin to emerge. And based on that, I developed this new theory or this new model of what we call aversive classical conditioning. Now, aversive just means unpleasant. And you'll notice that initially I talked about a foot shock, right? A foot shock or an electrical shock by definition is aversive or unpleasant. But even with this eye blink conditioning that I just talked about, again, if somebody blows a puff of air in your eye, that is aversive. It is unpleasant. Okay, so I can do this in a rat or a rabbit, and it is an aversive US. Now, in the rats, in much of my work, instead of using an air puff, we actually just administered a really mild shock right by their eye. It has the same effect. It leads the animal to blink, to produce a blink unconditioned response. And the animal can associate the CS and the US, and they show a conditioned response in exactly the same way I just showed you. But that's the important point, is all these learning paradigms that I've been talking about is based on this aversive classical conditioning. So what responds to aversive or emotionally arousing stimuli? The amygdala does, okay? So whether I was talking about context conditioning or eye blink conditioning, we know that the amygdala is involved in all of these various paradigms. And in all these various paradigms, the amygdala can associate the CS and the US, exactly as I talked about earlier when we talked about fear conditioning. Now, as I began to think through this model, there was an older model that's been around since the 1970s. This is called the two-stage model. And this was developed by psychologists who essentially argued that when it comes to sensory motor learning, again, responding to a sensory input with a specific motor response, this is challenging, right? Because you have to learn to respond in a very precise way. So it's actually kind of slow. It takes dozens or hundreds of trials to successfully learn. The fear conditioning, where I just present a CS tone and a foot shock US, that's really easy to associate and learn. A human or an animal can learn that in 5 to 10 trials. Okay? But sensory motor learning is just more difficult because a precise motor response is required. So if we go back to the idea of eye blink conditioning, I present a tone CS and then the air puff US. Well, because the air puff is aversive, that activates the amygdala. So the amygdala very rapidly associates the tone CS with the air puff US, and it leads to these fear conditioned responses: elevated heart rates, vocalizations, freezing behavior. But we also knew, based on this two-stage model, that it can accelerate or facilitate the acquisition of the sensory motor CR. Okay? So in other words, to come back to the example that I was just using, eye blink conditioning. So if I pair the tone CS and the air puff US, the amygdala associates those two stimuli very rapidly, and then it drives, it accelerates the learning in the cerebellum. It accelerates the association of the CS and the US in the cerebellum that leads to that conditioned eye blink response. So again, how do I know that? What's the evidence? Well, here's another example. Here is a control animal, and this is normal learning. So I'm showing you across training sessions, how many conditioned responses, how many eye blink conditioned responses did they produce? Okay. So if I present the CS 100 times, how many times do they produce an eye blink conditioned response? So early on, they're doing it about 20%. After 4 training sessions, they're doing it about 60%. So this increase is my learning. The animal has learned to associate the CS and the US, and they're producing an eye blink conditioned response. Now here's my experimental group, this one on the bottom, this line on the bottom, which is almost flat. This is an animal in which we injected a drug that kills neurons. So all these little brown dots you see, these are individual neurons. Here, if you can see it, this kind of yellow line, this is a needle that we inserted down into the amygdala. And we inserted a little tiny bit of drug, but that drug killed all those neurons. That's why you don't see any brown dots over here on the right side. Okay? So in other words, we damaged the amygdala. Now these animals are undergoing eye blink conditioning, but they no longer have a functional amygdala. So according to the two-stage model, we would argue that if we have no emotional CRs, then eye blink conditioning is no longer accelerated. It actually occurs at a slower rate. And that's exactly what we see, right? This line goes up a little bit, but not very much. Without an amygdala, without this early emotional learning, we have now decelerated or slowed the sensory motor learning, the acquisition and production of eye blink conditioned responses. Okay? Now that's what we knew up until this new model that I proposed. So if we go back to these 3 different areas, amygdala, hippocampus, and cerebellum. The amygdala is responsible for emotional learning, right? If there's fear involved, that requires the amygdala. The hippocampus is involved when you're encoding and learning about new contexts, new locations. And the cerebellum is involved when there's a sensory motor response, a very precise motor response is required, such as closing your eyelid at the correct moment to block that air puff. Okay? So what I argued is that all 3 of these brain regions are interacting during eye blink conditioning. And they're interacting in a very specific way. Okay? Now, I need to step back again. I've talked about the idea of synaptic plasticity and associative learning. So when I said that there is an increase in associative strength, that's what leads to the production of a conditioned response. But again, based on many decades of research in behavioral neuroscience and learning and memory, we know that there's not an infinite amount of learning available or associative strength. The amount of associative strength in the brain, that is how many CSs and USs can be associated together, it's not infinite, it's finite. Okay? So here's the idea. Early on in eye blink conditioning, what's activated first is the amygdala, is associating that aversive air puff US with the CS. The hippocampus is associating the context, the location, the conditioning chamber when this is happening. So right here at the beginning, there's a lot of the two grays. That's because most of the responding on the part of the animal is being controlled by the amygdala and the hippocampus, the fear or the emotional CS-US association and the context US association in the amygdala. Okay, so we see lots of dark gray and light gray. This is the behavior. This is the freezing behavior I talked about earlier. But as we pair the CS, the tone, with the US air puff, again, over time, the animal begins to learn that association. The animal begins to emit more and more conditioned responses. That's what this white here is. It's an increase in eye blink CRs, eye blink conditioned responses. But again, there is only a finite amount of associative strength. So as learning occurs in the cerebellum, as more and more eye blink conditioned responses are emitted, there must be a corresponding decrease in the amount of behavior controlled by the amygdala due to the emotion or the hippocampus due to the context. So at the end of eye blink conditioning, we have a lot of eye blink CRs being emitted, but very little fear responding due to the CS and very little fear responding due to the context. All right, last couple of slides, folks, and I'll finish up. So here's my model. We have rapid emotional conditioned responding early in the amygdala, which leads to the acceleration of this sensory motor learning, the eye blink conditioning. But remember that the emotionality of these animals is elevated, it's heightened, right? Their fight or flight response has been activated. So what is required is a mechanism for the cerebellum to inhibit the amygdala and those emotional responses. This is the third stage that I propose, emotional CR suppression. You have to suppress the emotions as a result of this learning. So to quote myself, the model provides the immediate benefits of conditioned emotional arousal on sensory motor learning, while also ensuring emotional reactivity, high levels of emotional responding, that it is ultimately stabilized and returned to baseline levels. If this does not happen, so let's imagine we have somebody who suffered a TBI, a traumatic brain injury, or maybe they had a stroke and their cerebellum is damaged. Okay? Their cerebellum is impaired, but we want to do eye blink conditioning in there. So we pair that tone CS and the air puff US. Their learning is different from our control, our quote normal person. The normal is shown over here. Okay? This is what we just talked about. The emotional responding via the amygdala and the hippocampus, that's activated first very rapidly in stage 1. That leads to the acquisition of our eye blink CR, our eye blink response due to the cerebellar learning in stage 2. But as the eye blink CRs grow in number, they simultaneously inhibit the amygdala and this emotional responding. That's why the box is smaller here and the font is smaller. That's because we have a suppression or an inhibition of that emotional responding. Over here is our patient that has a damaged cerebellum. Okay? So the emotional part still occurs. The amygdala and the hippocampus are activated and we have high levels of emotional responding. But because the cerebellum is damaged, we don't have much learning occur. There's very few eye blink CRs, and there's very little inhibition of the emotional responding. So in other words, emotional responding, that elevated, prolonged, heightened state of emotional reactivity remains elevated. It's no longer being suppressed or inhibited as would be required in the 3-stage model. Okay? And this is because, again, the cerebellum is damaged. Now, is there any evidence for this? Well, not much. Again, this is a theory or a model, but it is known in psychiatry that patients that have cerebellar damage, again, due to a stroke or some other biological reason or external causes, a car accident, or maybe you're a veteran, a combat veteran, whatever it is, your cerebellum is damaged. They do typically have an increase of anxiety, right? The two of those are correlated, cerebellar damage and anxiety. So why do they show anxiety? Well, again, to finish up here, if emotional CRs are not suppressed, the subject will experience a prolonged heightened state of emotionality, which over time may then generalize to a mood or anxiety disorder. So if the subject is all hyped up, right, they're feeling their fight or flight response activated, right? They're gritting their teeth, they feel their heart rate beating fast, but they don't know why. Over time, that can generalize to a state of anxiety on the part of the patient. So anxiety is a apprehension or fear. Sometimes we know why we're anxious, sometimes we not. So this would be a case where the anxiety, you may not be able to identify the reason, but the anxiety is still there. So just to finish up this concept, I've talked about the idea of knowledge for knowledge's sake, that is understanding. I've talked about trying to apply that knowledge to treat or understand different disorders, such as fetal alcohol spectrum disorder. With this new model, this three-stage model I've developed, I've tried to offer some ideas in terms of treatment as well. So again, if you'll allow me to quote, such an account, like this account that cerebellar damage may be responsible for heightened emotional reactivity, which then may lead to anxiety. So the anxiety is due to cerebellar dysfunction, okay? That account diverges from most neuropsychological models of mood and anxiety, which generally focus on the amygdala and related limbic structures. So the limbic is just emotion. So these are the areas of the brain involved in emotional reactivity, the amygdala as well as the cerebellum, or excuse me, apologies, the amygdala and the hippocampus. So a consideration of emotional dysregulation from the perspective of the cerebellum may therefore help reframe the conceptualization and treatment of certain psychiatric or neurological disorders. So I do hope that neurologists, psychiatrists, if they become familiar with this model, again, it may offer an alternative view, a different perspective to understand why cerebellar damage could contribute to anxiety. So I know I've gone over a few minutes, I apologize, but to finish up, let me leave you with a quote by this guy named Louis Brunel. He was a Spanish artist and filmmaker of the mid-1900s. I love this quote because it really gets at the importance of learning and memory. You have to begin to lose your memory, if only in bits and pieces, to realize that memory is what makes our lives. Life without memory is no life at all. Our memory is our coherence, our reason, our feeling, even our action. Without it, we are nothing. Again, memory is what allows us to reflect on our past, to adapt to our current situation, and to make predictions about future events. It's absolutely critical to the survival of all organisms, including humans. So with that, thank you all very much. I appreciate this opportunity to come speak. And with the time we have remaining, I would be more than happy to answer any questions.
So yes, we open it for discussion. If you have any questions, please feel free to ask. I know there were a lot of concepts covered in Dean Derick's presentation. So you can start from any basic question to a complicated one. Every question at this stage is pretty valuable.
Yeah. And again, I will be honest, again, I went into this talk recognizing it was a challenging talk. So I hope I didn't turn any of you off, but I really wanted to show, again, what is behavioral neuroscience? What do we do? You just don't see that in India. So I really wanted to kind of illustrate that for everyone out there. About the anti-inflammatory, the ibuprofen. Yes, Maliha. So if I could just jump over here. So would it help combat or counteract the effects of FAS? It could, in theory, okay? Now, I don't recommend that any pregnant women, have a six-pack and then take a couple of ibuprofen. Ibuprofen has its own negative effects, particularly when it comes to a developing fetus. So our study was more of a proof of concept. that by tamping down or diminishing the inflammation, that can be beneficial for the developing organism, but there would be much more work required before we could ever make this available as an actual treatment for women. And there is the added danger that if you don't simply try to discourage drinking during pregnancy, if you say, here's an alternative, then women may drink. Personally, I don't buy it. I think if there's a way we can help, we should do it. It's like people that are anti-abstinence or abstinent pro, I guess, right? We are going to talk about contraception because then people might have sex. Well, people are going to have sex anyway, right? Women are going to drink anyway while they're pregnant. If there's something we can do to help them, we should do it.
Thank you for that. Yes, more harm reduction, I think you're speaking to, rather than complete prevention, more harm reduction.
Absolutely.
Potentially.
Never claim it would be 100%. And one of the reasons we work with the rodents with the third trimester is because our animals really have no physical effects. They grow up looking perfectly normal because your body's developing during the first and particularly the second trimester. That's when the body comes online. By the time you get out to the third trimester, which is when we're administering the alcohol, that's really brain development that we're affecting, but we don't see the physical impediments in our animals, which makes it easier to test them and whatever results we find to contribute it to alterations in brain development, then again, some sort of physical impairment.
Thank you.
So I have a question. I saw the model which you proposed at the end, and I know to a large extent, it's stemming from your work, and especially the last part, it's based on the understanding. How do you see moving forward, this can be tested both in animal research as well as in human research? Because the implication is trying to grip that cerebellum is possibly involved, and that is associated with generalized anxiety and depression. That's quite a big implication, has a great impact, how these disorders are treated. I just wanted to understand, where do you see the next steps going forward?
Yeah, great question. Thanks, Kanika. So, I mean, I didn't develop this idea out of the blue. Over the last probably 10 or 15 years, there's been more and more research showing that the cerebellum is not just involved in movement. It actually does play a role in our emotions and even our cognition. So there was already speculation out there that the cerebellum could be involved in anxiety and other disorders, but there was no real model to account for how cerebellar damage could lead to persistent elevations in emotion. So I think that's one of the benefits of this model. And I actually, in putting and writing this paper, I found literature that was almost 20 years old, where multiple authors had damaged the cerebellum because they were trying to understand, was it involved in eye blink conditioning? And they say, look, if we damage the cerebellum, those eye blink conditioned responses disappear, right? No surprise, that's what they were trying to find. As an aside, they would go, oh yeah, we also noticed that these emotional CRs remain elevated across conditioning. They didn't make a big deal out of it. It was just kind of a passing statement. But as I began to get into this, and I thought, well, is there any actual empirical support for this idea? If not, it's not a very good model. But I was actually able to find some old studies that do offer support for the theory itself. Now, that said, it's still very theoretical. I think everyone knows I'm no longer doing this type of research, so I'm not going to prove that it's correct. But, hopefully it maybe a few people read it here and there, and it will sort of catch on, and people will take it more seriously as we move forward into the future.
Thanks, Derick. There's also a question from Zaraksh. Do you want to unmute yourself and ask the question? Otherwise, I can read from the screen. I believe the most commonly used general anesthetic interact with Nmda system. What effects do they have on memory and learning?
Yeah, so the Nmda system, you are right, can be activated through anesthetics, but the more problematic are what we call Nmda receptor antagonists. So an antagonist is a drug that blocks the action of Nmda receptors. One of the most, or the two most popular are PCP, psilocybin, if you've ever heard of angel dust, or ketamine, which is a psychedelic. So both of those at low doses induce a psychedelic trip in the people. At high doses, they have really bad effects. They can really mess a person up and even be fatal. But let me try to put this in a little more concrete terms for you. So if you drink a lot, if any of you have heard of the term a blackout, what is a blackout? A blackout occurs because you have so much alcohol in your system that you are essentially shutting down the Nmda receptors in your hippocampus. Without the Nmda receptors, you can no longer encode what's happening to you at any given moment in time, i.e. you can't form new autobiographical memories. So the next day you wake up and you're hungover and you've had a blackout, you don't remember 3 or 4 hours of the previous night. That's because that memory is simply not there. By blocking those Nmda receptors, you have essentially hindered the ability for that autobiographical memory to even be formed in the first place.
So in terms of reading, we will be sharing, I think that's why we requested everyone to register. Professor Derick has already shared with us some pre-selected reading, and we will be sharing that with you all.
All right, well, thank you all very much. And again, for those that stuck around, I appreciate it. Again, I know this is not a normal talk for JGU, so I hope that at least you understood parts, and if nothing else, you enjoyed the topic and you learned a little. So thank you, everybody. I appreciate your time. Have a great weekend.
Thank you, Derick, for such an informative talk, and talking about a topic, as you said, it's not much, there in Indian scenario. And I hope it motivated a lot of faculty and students. And I hope the channel of communication we open this channel of communication, and they can reach out to you if they are interested in expanding this field or learning more about that field. Thank you so much for your time and kickstarting the event.
My pleasure. Thank you, everybody. Have a great night.
Bye. Thank you, everyone.
All right, well, there we go.